Wait, Jackie, aren't you on vacation this week?
No, just taking the day off tomorrow, but
Okay,
that'll feel like a vacation.
fair enough. I love that you're going to be hanging out with Sarah. Such a small world.
Yeah, I know. I'm so excited.
I'm just going to give people one more minute. And today, Samreen is going to be presenting the paper that we chose on multi-generational effects in Daphnia magna. So, looks like she already has her presentation up. Oh, John, I think you need to mute yourself.
Okay, one sec.
Oh.
दो मिनट में
All right, it looks like we don't have anyone else coming into the waiting room. I'll keep an eye on that. Samreen, I'm going to go ahead and let you take it away. The recycling truck is about to arrive in front of my house, so it's getting very loud. So I'll let you introduce yourself and the topic.
So, hello everyone. This is Samreen Siddiqui, and I'm a postdoc scholar at Dr. Brander's lab. I'm presenting the paper already submitted to you, already shared with you. This is a multi-generational paper on Daphnia magna, and let me take you directly to it. In this paper, the major points they have is microplastic aging. And they said like aging can change the physiochemical properties, as we all consider and think appropriately about UV and other effects. Second point they have is they have a comparative data on effects of age compared to pristine microplastics. And that is scarce, and that's true. The data is really scarce, and there is still like a lot of research going on, but still not to the point that we can say that we have enough data. And third point of this paper, they said one of those aging process is absorption of chemical. And that really has a scarce data about the absorption of the chemicals within the microplastics. So basically, they took four concentrations, 80, 400, 2000, and 10,000 particles per milliliter, and compared it and they kept these particles inside the wastewater and in the ultra pure water. When they kept it inside the wastewater, they were like there for I think for 1 day or 2 days, but I have more description later on. Then what they did is they kept Daphnia magna over 4 generations. Another thing they added is they limitized its food, and they said like that is what we expect into the environment, which I have a point of discussion at the end. So basically they decrease their food level and they put them inside into the wastewater and ultra pure water, and basically make them to eat those plastics until 4 generation. So they prepare their own polystyrene microplastics from the coffee to go cup from the local bakery. What they did is they just cut the lid into the small pieces freeze dried in the liquid nitrogen, and just grind it through a swing mill. The powder they sieved it through less than 63 micrometer size sieve, and then that's what they use for the experiment. In their water preparation, they sampled the influent of wastewater treatment from Germany. And since this paper is in Germany, the effluent they used is raw wastewater, and that is treated with bar screen only. So only solids are out. Most of them is liquid. That's what absorbed into the microstyrene particles. The 24-hour composite sample that consists only 1 liter of wastewater sampled every 2 hours. And that's what done in between March 31st and April 1st of 2019. In addition to that, there was no rainfall recorded in those 2 days. So that's just the concentrated wastewater. That's what they used for the polystyrenes. Wastewater actually filtered using 0.2 millimeter filter through a rapid flow Nalgene filter. And after that, just samples were removed and suspended into the solids and microbes. So it contains microbes as well as solids in there. The MP, then the microplastics then incubated in the filtrate for 1 gram per liter at 4 degrees centigrade up to 38 hours. So around 1.5 days. After the incubation, it was recovered by vacuum filtration using a 0.2 micrometer filter again. Those particles then froze at minus 80 degrees and lyophilized to remove the residual water. So just basically freeze dried. The pristine microparticles were treated identically, except that they were incubated in ultra pure water. So basically what they did is they treated the ultra pure water same way as the wastewater, kept that there for 38 hours at 4 degrees centigrade and just freeze dried it. The endpoints they reported is survival, reproduction, adult and neonates, body length, and growth. And as per their results, survival for 3rd generation, so for F1 generation, is actually got missed here at highest concentration. That's microplastic at 10,000 concentration in wastewater treatment plant. Actually went higher, whereas waste in ultra pure water that just got extinct at in this F2 and F3 generation here. So, and basically they found most the survival going down with pristine microplastics when compared to wastewater treated plastics. So if they compare between them two, in F0 generation, it's almost similar, but as you move to the F2 and F3 generation, the highest particles without wastewater showed most impact. In the reproduction, in case of reproduction, again, microplastics, pristine microplastics at 10,000 concentration, that's where the offspring actually went down. And by F2 and F3 generation, completely extinct, because they can't find any reproduction at F2 generation. So F3 got extinct again. In other, they don't find much effect. In other exposure group, they don't find much effect except this one. When they compared the body length, again, at 10,000 microplastics in F2 generation, they found the body length like very low, or because there was no survival, they just can't find the body length. And that's what they said, actually, that get decreased as well. Whereas in F1, F0 and F1 generation, we can't find much difference. When they compared the body length, they found a concentration dependent effect on the body length development over the time, over the 21-day period. Again, this is pristine microplastics, and this is wastewater incubated. And there were not much effects, like differences seen between the two. So basically what they said, that an exposure to pristine microplastic actually resulted in toxic conditions and extinction of 3rd generation in Daphnia. whereas the wastewater incubated particle actually increased a lower mortality. So that means it's a healthy water. And one of that reason is because of the nutrients content within the wastewater. It may help Daphnia to grow further and compared to the pristine microplastic where there was no nutrient in the water at all. So overall, the weakness I found in the study is they limitized food and they said it's environmentally relevant condition when they are exposing at two different media. So one is wastewater, nutrient rich, and they are reducing the food content, and another one is just ultra pure water. And again, they just limitize the food, which I think was not appropriate. Second thing I found is it looks like studies promoting wastewater effluent to be a good source when it is not, and that's what they don't want to say, but it looks like. An exposure method that could be a caution, which we can discuss after this. The strength of this study I found is it has a multi-generational approach, so which is very like not much studies do that, and it's very important. And it is interesting approach how they integrate microplastics with wastewater. So they let that absorb for some time and then they used it. It's like very few studies that does that. And at the end, I would like to bring up some discussion questions, what you all think about this study. What causes two incubated media act differently? Maybe you have some interesting points. Second point is temperature. We may have some effect on incubation at 4 degrees. I don't think it may absorb much, or if it does, how it can differentiate between the two. And chemical analysis in wastewater. I think there is something else other than just these chemicals into the wastewater that can cause more fluctuation in this study. And as I pointed out, nutrient, I can think of one. So that's what I have today to present, and we can discuss on these points if you want.
And Samreen, since we have some new people in the group or people who haven't joined us in a few meetings, it might not be a bad idea just to go around and briefly introduce ourselves again, since we haven't done that in a little while. I mean, I can start. I'm assistant professor at Oregon State. Ecotoxicology, microplastics, and pesticides are what encompass most of our research. And Samreen, you introduced yourself in the beginning. So maybe just go around the room and have people give a brief blurb about who they are and where they're coming from. Jackie, can you go next?
Yeah, sure. Hi, everyone. I'm Jackie Ling. I'm a PhD student in agricultural and environmental chemistry at UC Davis. And I'm interested in tire wear particles and how aging processes of tire wear particles affect chemical leaching over time.
John, on my list here.
Hey everyone, I'm John. I am a master's student in Dr. Brander's lab, and I also do research on tire particles in addition to a bunch of other different plastic kinds, and looking at the toxicity to estuarine indicator species.
All right. Ariadne?
Hi, everyone. My name is Ariadne. I'm actually in Maine right now. I'm on the East Coast. I am interested in making packaging that is ocean compostable, fish friendly. I am a marine scientist, graduated from the University of Maine here, and now I've looked at reviving a heritage industry in New England of pulp and paper. And so I'm really excited to see the research. I've been introduced to this group through Tracy Harper, Stacy. I do have a Tracy I work with here, but Stacy Harper, who I'm excited to work with more and see what you guys are doing with how you test the packaging. My whole hope is to make a standard that any packaging can strive for that's ocean compostable, fish friendly. And right now I'm unpacking what that means and how to quantify that. And it's probably the company's Paramount Planet product. And we've been doing it for 2 years, and we are supported by a National Science Foundation Small Business Innovative Research Grant, phase 1, and now we're applying for our phase 2 right now. So thanks for welcoming me to your group, guys.
Carrie?
everybody. My name is Carrie Sand. I'm an assistant professor at San Diego State University. I'm a toxicologist, but I focus mostly on human health and risk assessment and human toxicology, namely endocrine disruption and reproduction, developmental toxicology. But I've been working with plastic chemical leachates for like a decade now. So even though I didn't really jump in, I know a lot of the conversation is surrounding particle toxicity. I've been working with things like phthalates, surfactants, things like that are the additives for a long time, and they're a very big passion of mine. So very happy to be here and hear from all of you. Thanks.
Scott, looks like you're up next.
Thanks. I'm a research scientist at the California State Water Resources Control Board, and I'm researching the impacts of microplastics on humans and ecosystems and developing some health-based thresholds with Suzanne.
Paul?
Hi. I have to say, Suzanne, I like wherever you're sitting. That seems like a really perfect spot to have your meeting.
Now that it's finally stopped raining, as of yesterday, I'm out here all the time. So, yeah.
Yeah, good. Hi. My name is Paul Meyer. I'm a research ecologist with the U.S. EPA Office of Research and Development in Corvallis, at the lab in Corvallis. We are just beginning some work to try to understand and characterize tire wear particles, and to potentially use green infrastructure to capture tire wear particles, as well as 6-ppb. We're just in the very infancy of that work and trying to catch up with you all who've done a lot more work than we have. And so I'm just I'm an observer and a very keen listener here. Thanks.
Violet?
Yeah, hi everyone. I've missed the last couple of meetings, but I'm happy to be back. I'm a senior scientist for the Orange County Sanitation District. So yeah, coming to you from a wastewater treatment plant. I actually did not realize the topic of the paper until this morning. So really interesting stuff. I'm the microplastic lead here at OC San, and we're working on a couple different microplastic projects, including some with the Southern California Coastal Water Research Project, or SCORP, I guess my background is in toxicology, marine ecology, and a little bit of chemistry, and just happy to be here. Happy to see you all.
And Brittany, last but not least. Okay.
Thanks. Hi all, I'm Brittany. I'm a PhD student in toxicology at Oregon State University, and I study effects of micro and nanoplastics on small aquatic organisms.
Cool. So Samreen, I'll let you take it from here on leading the discussion. Maybe we can start with your first question, which was definitely on my mind when I was reading the paper. What were your thoughts on the differences between the two media that were used?
Frankly, I think it's wastewater, it's nutrients, full of nutrients. That's what causes them to increase growth. And ultra pure water missed all those things. In addition to the chemicals, I don't know how well they absorbed at 4 degrees centigrade. That needs to be looked into. So, but I think there are so many things need to be looked, like what chemicals in there, what causing these changes, temperature or whatever. That's what my thoughts anyone has different thoughts on it.
Yeah. I also wondered how the 4 degrees Celsius, that incubation temperature, was affecting the ability of the chemicals to absorb. And also the incubation was relatively short too. So was there really time for adequate absorption? I agree that there seemed to be a lot of information kind of missing, a lot of assumptions being made. And I know that the study was a lot of work. Rearing animals through 4 generations is, and all the endpoints that they looked at, that was a pretty intense amount of work. But I agree that I thought that if they really wanted to answer the question of how wastewater might be differentially affecting the response, we needed some more information about what was happening as far as the chemistry of those two different media goes. But you know, what did other people think? I guess Violet, you might be at the closest to this. To this not to single you out, but
No, it's all good. I'm happy to weigh in. I thought this was really interesting. Like I liked actually the design. I had all the same questions, so I'm glad you guys brought those up. And I don't know if they justified the 4 degrees anywhere. It didn't seem like it, right? And so I like part of me was just like, is that just to prevent like smelly decomposing raw sewage odors? I don't know. I would have the same thoughts. I thought at least like, you know, there's some missing components and definitely some follow-up questions. I thought the design overall was pretty elegant though, like it generally. I also thought it was interesting that they basically were like, yeah, we know it's important to look at what chemicals might be causing this, but there's so many chemicals so we're just not going to do it. Which I guess I would love to use that excuse more.
I'm not sure how that works, but I'm glad.
Yeah, I think, you know, from my perspective, one of the things so I don't necessarily have good answers to any of those observations other than I agree. But I think something that struck me was that I would think for this to be more relevant, you know, I would have looked at a secondary effluent or a treated effluent and not so much a raw sewage, because like Daphnia, I don't think are really going to be living in that raw sewage. And it's the treated effluent that's going to be, you know, where critters are living and where that, you know, the effluent's going to be discharged into receiving waters. So I think it's like a good start because it's interesting to see it like the highest strength organic matter. How does that interact with some of these parameters? But I'm not sure how relevant that is to organism exposures. That was one of my thoughts.
Yeah, I agree on that. And it would also be interesting to not just look at treated effluent, but to compare different levels of treatment. So secondary versus tertiary, for example. Although this was published in Europe, so they might do all tertiary there. They might have that same concern.
That's a really good point. That's really relevant, especially because they're here in the United States, there's a big split between agencies that do different levels, right? And I think it all comes down to, you know, how much solids are removed, right? And so you're comparing, you know, the amount of organic matter in those samples.
Totally.
So was the temperature selection, they didn't justify it. Did it have something to do with supporting Daphnia?
No, because the Daphnia exposures were at 20 degrees. I think they're pretty temperature sensitive, right? And I used to be a Daphnia mommy in a past life. I used to do this work.
Yeah.
I feel for them.
Take care of a room of Daphnia.
So you know, dissolved oxygen is going to change with temperature. So there'll be more dissolved oxygen at 4C. I'm also wondering, you know, about light exposure and just, you know, if they were controlling for that, or if that is also a factor in decomposition or in transformation.
I guess, well, they didn't really look at it after the treatment process. If they were looking at effluent after it was treated, light might not be that much of a factor during the treatment process itself. But yeah, that's a good question. That would definitely be relevant if you were trying to think about how particles would behave or act in terms of toxicity after they've been in the environment. So yeah, it's a good question. I'm not sure.
Yeah, and I'm also not sure why they limitize the food. Like what make them to limitize food? Because that means you are making them to eat those things.
I was a little bit surprised about that too. I guess maybe, and they didn't really explain this, but maybe they had the high food treatment versus the low food. Maybe they were trying to think about a seasonal comparison where you
They
would,
just said that's what they found in environment.
yeah.
I'm blanking on the paper I read this in, but I do remember reading a different one that was a plastic exposure where they like didn't see any effects until they limited food. So that came to mind when I first read this, that perhaps they've tried with like the standard amount of food that you would give in a lab experiment and they didn't see effects, and so they decided to limit the food.
I'll chime in. So I work with like things like phthalates a lot, for example, and I know because a lot of them are really good PPAR signaling pathway agonists, right? It causes them to eat a lot more. So I know, for example, like if I don't do the high food challenge or something like that, either I get early or like you said, like it just doesn't appropriately allow the experiment to proceed sometimes. So I just assumed when I was reading this that was the reason for that. But good points. I wasn't thinking about it from the other perspective too.
In this figure 2, they did high food and low food component, and their low food is actually 100% survival, and high food is just about 80%, which is, but in generation 1, they have a good survival at high food and so on. So something happened only in generation F, not generation, that makes.
I was just going to say that might just be sort of your expected variability in survival over a longer term test. I know for regulatory purposes that for a chronic test, 80% survival is kind of the acceptable survival level. But yeah, it is a little bit. It's interesting that they had that much of a difference.
What did you guys think about the improvement in survival for generations 2 and 3? Like for I guess both colors, both wastewater and the other ones. I thought that was pretty interesting. As the takeaway, what I got from that is the potential for, acclimatization.
I thought about that too, and that had me wishing that they had looked at some molecular endpoints, like DNA methylation, for example. Although I did investigate, and the genome for Daphnia magna just came out at like the end of 2019, so they may have not had the information they would have needed to look at that. But it does make me wonder if there were genes methylated or differentially expressed due to the exposure in that first generation or two that then facilitated sort of a rapid adaptive response in the later generations.
Yeah, because it's a pretty impressive jump to go from, 20, 30, 40% back up to control levels. And those are pretty high concentrations, especially at the non-wastewater particles. They pooped out completely. I shouldn't use that word. Maybe I should.
Yeah, but they don't have even a standard error if they are significantly different or increased or not. So that's a missing point here.
I didn't catch that. That's a really good point. I wonder why, because they had a lot of animals.
Yeah.
I would have also liked for them to fit those response curves instead of using our plots, but all the information is there at least. But yeah, I agree. Where's the standard error?
I was wondering, this is just sort of an aside, but what everyone's thoughts were on considering the ground up coffee lid plastics as like pristine plastics. Is that standard? Would you consider those to be pristine? Like, I guess I normally think of like pristine microplastics or pristine plastics as like pre-production pellets that haven't been processed. I would consider these to be more like post-consumer. I don't know. Am I like reading too into it?
This
I guess I was
question,
curious.
this question is very important to me. So I'm really glad you asked it, actually. The whole difference of figuring out if it's weathered versus virgins and the data that's behind it. Specifically, coffee lids. So because that's one of the things we're looking at making. So.
I guess maybe we're getting it to a point where the definitions for some of these things are shifting a bit. And I agree, it does seem a little bit fuzzy, and it does seem like pristine, at least it used to mean pre-production or primary, you're thinking of nurdles or, you know, uh, maybe even the plastics used for three-dimensional printing, like the true primary plastics. But I can understand why they're calling it pristine, because it hasn't been exposed to the aquatic environment yet. So, but yeah, the definition is a little, it feels like that definition is shifting a bit here.
Yeah. Yeah, it's like non-weathered versus weathered, or non-exposed versus exposed. I think that to me there just could be a difference in the other chemicals or additives that are added to that plastic cup lid, stabilizers or anything else that could actually be leaching out or contributing to responses here that might not otherwise be. But at the same time, that's more probably realistic to environmental exposure. So I was just curious about the terminology there.
Yeah, I guess if they had called them virgin microplastics, I would have had an issue with that, because to me that means you've removed all of the other additives and potential associated contaminants. Whereas here, pristine, I guess makes sense only because they're comparing something that's been weathered. But yeah, it feels like
that makes sense.
It feels like we need a new glossary. The definitions are shifting for sure.
Yeah, that's a good point.
Samreen, what were some of your other questions?
I think I covered them all.
Okay.
Yeah. I covered most of them.
Yeah, and the chemical analysis question is difficult. I mean, really, to identify all of the chemicals in that in the wastewater, they would have had to do some sort of non-target analysis, which is almost another paper in and of itself, almost. Yeah. But yeah, in terms of, did this study really realistically create particles as they would have been, coming out of the wastewater treatment process, I would say probably not, given that they looked at raw sewage or pre-treated water rather than post-treatment.
Yeah, that's something I think like really important, because there are very few places if raw wastewater has been flowing into the river in case of maybe flood or something, but most of the time it's effluent which goes.
And that might actually help answer some of these questions if you did look at other types of effluent in understanding, is it the degree of organic matter present, or like what is it about that sample? I think to me, that was the other thing that was missing, is that they looked at sort of the two extremes, right? Ultra pure water and then raw sewage, and there's lots of like more relevant water media in between, like just surface water, or even tap water. That might have some more constituents in there that could alter some of these responses.
And they limitize the food. So they are not getting nutrient, and they limitize the food. So it looks like they skewed their results.
Yeah, that's a good point.
To death. Anyways.
Yeah, that would not be a nice medium for them. Just ultra pure water with no food. That doesn't sound like a nice, happy Daphnia home.
Mm-hmm. Yeah, and another consideration would be, and I'm thinking back to work I did in consulting when we were testing effluents, treated effluents from different wastewater treatment plants in California. And we would always do kind of a dilution series because we were trying to take into consideration that the water it wasn't just going out as pure wastewater, it was immediately being mixed with surrounding water in an estuary or coastal habitat. And so looking at diluted effluent, a gradient of diluted effluent would be relevant here too. But then of course that's a lot more animals and time and money and all those things. But
yeah, I've been thinking about why they chose, we were talking about this a while ago, to incubate at 4 degrees Celsius. And I'm wondering if, at least from what I've seen, for the most part, hydrophobic chemicals sorb more effectively at lower temperatures. So I'm wondering if they were trying to maximize that sorption. But then it sounds like they're putting those microplastics in water at 20 degrees Celsius for the DAFNet exposures. So they didn't see necessarily toxic results from sorption, it doesn't seem like, and desorption, but if they had, I would wonder, is some of that chemical that was originally sorbed at 4 degrees Celsius just going to come right off in the water at 20 degrees C and kind of confound your results?
No, that's a really good point. I didn't think about that. That's a really good point.
I don't think they were thinking at chemical level because they didn't mention it anywhere in their whole script. I think they just chose 4 degrees because in Germany it's cold. So they were just trying to mimic the environment over there.
Not 4 degrees Celsius all year round though, right?
All the time we use those March and April, maybe. I'm not sure.
John or Brittany, did you have any thoughts about the paper in terms of, what we're doing in our own experiments here?
I think it's really interesting, especially because I just came from a conversation kind of planning for the summer because we're hoping to start moving into co-exposures, and we're having a discussion on whether to pre-incubate the plastics with some, say, pesticides we're looking at, or whether the exposure should be done together. And so, yeah, this was an interesting paper to read and see how they did sort of a pre-incubation and whether we wanted to do something like that and see if there is a difference if just the plastics are exposed and what they might sorb versus what the organism could take from the water. So it's given me a lot of thought moving into those similar types of experiments. It would be interesting to do it long term as well and look at those multiple generations.
Yeah, agreed. I feel like comparing pre-incubation to co-exposure is really important because you're going to have both situations in the environment. So, but yeah, and we've also been thinking about moving into multi-generational experiments. Probably not with fish this time around, since that takes forever. But maybe with mysid, our mysid shrimp model. So. We should do some side-by-side Daphnia and mysid shrimp multi-generational.
Yeah, that would be great.
So what size plastics would you look at for those, Suzanne, for the mysids? Like nanos?
We've been comparing micro and nano size fractions with Daphnia and mysids here. Daphnia in the Harper lab and mysids in my lab. And the microplastics are between 1 and 20 microns in size, and the nano are mostly just under a micron, so larger nano fraction with a small number in the smaller fraction. And they take up both. At least for the microplastics, we're able to clear the tissues and then look at the internalization of different particles. And they take up particles up to the 20 micron size. I mean, they will eat brine shrimp when they're just a few days old, and brine shrimp are 120 microns on average in diameter. So they take up particles larger than I was even anticipating. So yeah.
I do like the multi-generational component there, and I think that's so important, and especially with just how what's the word? I don't know, like inconsistent the toxicity responses seem to be to microplastics. It's hard to know, what the mechanisms are, and if you can get at that as much as possible, and especially considering organisms that are exposed for multiple generations, what does that mean? But that can help potentially elicit some of the mechanisms at play there. And it's very relevant, can they adapt, because this is unfortunately the future. This is where organisms are exposed to now, and I think it's safe to say it'll be like that for a while, whatever conditions there are currently there.
Yeah, absolutely. And what this paper shows is that there maybe is this ability to adapt, and potentially rather quickly. It would just be interesting to know what molecular mechanisms are at play there. What's driving it?
Yeah.
The nice thing about this model and other invertebrate models is that you can actually get at answering that question. It doesn't take 2 years to get to the F2. So that's makes it much more attractive. But yeah. Is there any burning questions that anyone had that we didn't address or talk about? No. We answered everything.
I just have a quick question about the next paper. Do you send that out before the group meets, or do we decide on it now?
We have been sort of just deciding as a smaller group, or someone will email me with a suggestion, and that will be the paper for the next month. So we don't have a super formal process for that, but if you have a paper or a topic area that you are super interested in, I'm happy to take suggestions, or if others have suggestions for the next meeting. We might end up skipping July for the FX group because we have a teleconference around the same time. I think it would end up falling on the same week, but then take things up again in August. Also, a lot of people are out of town in July. But if anyone has suggestions for the August FX group meeting, happy to hear them. What did you have something in particular you were thinking on?
No, I just want to know if this is the format of the group. We read a paper and then we talk about it next time.
Mm-hmm. Yeah, and I usually try to send the paper out at least 2 or 3 weeks ahead of time, so people have a chance to read it beforehand. Wow.
This group, I've been doing the communications group, and then I found out at one of the last group teleconferences, that this group is also going to. So really cool to learn.
Mm-hmm. Samreen, when you were doing the background reading for this paper, did you come across any other similar studies in the literature? There's not a lot on.
Not like this kind of where they've solved. Like there are other kinds of weathering going on right now. People are doing several UV things, but not sorption. Here is another paper I seen today, this morning, is doing similar kind of stuff, but they are doing at various pH. So, and they are not looking at the organism, they are just looking at like how much they sorb. So different people are looking at sorption mechanism, but not in multi-generational study.
Yeah, I want to say I've seen a paper or two in zebrafish, but it was not looking at, or at least attempting to look at a more environmentally realistic scenario. So this paper seems to be kind of the first of its kind, at least.
Yeah, but even in this paper, there was not significant results seen at environmentally relevant concentration. The only significant results they seen is at 10,000 microplastic concentration. So it's way out of environmentally relevant.
Yeah, I think I meant more in terms of that they compared weathered plastics to non-weathered. But yeah, agree that even their low concentration was pretty high. It was 80 particles per mil. So
yeah.
Mm-hmm. Yeah, I mean, another really interesting thing would be to compare maternal to paternal exposure. Of course, you couldn't do that in Daphnia easily, but in an organism that has clear differentiation between the sexes. So you could do that in a fish species or in a different invertebrate species potentially.
So I'm not sure if this is quite so, because I like to look at paper-based packaging, and one of the problems is PFAS, which I don't know if it's as prominent in plastics, because like there's other toxic chemicals that they absorb, but so I don't know if that's within the scope of this group, but maybe we could look at some papers around those effects, especially in wastewater. I know there's a huge problem.
Yeah, I think that would be really interesting, especially maybe in terms of fibers. I'm thinking of all the clothing that is treated with PFAS to make it wrinkle-free and stain-free and, indestructible. So, yeah, totally. I like that idea. Well, I think we maybe are going to finish up early today unless anyone else has something they wanted to critique or talk about in terms of this paper. I mean, I think it sets a really good kind of baseline for doing multi-generational studies with microplastics, and it's something solid to build from, for sure. And I hesitate to be too critical because it's the first time someone's attempted to do something like this. So it's easy to poke holes, find all the little holes in a paper. But it was a well-designed study, and definitely, I'm sure there'll be more to come like it. But, yeah. And Violet, I appreciated having the wastewater perspective. So thank you for joining us today.
Yeah, I'm happy to be here. And yeah, I agree with you. I think it was a pleasure to read and to think about. And I think, like you said, I think it just gives us a lot to think about, and continuously improving study designs and making them more relevant, and really trying to understand the mechanisms of what's going on in the real world, right? And not just in these lab exposures. Yeah, always happy to talk wastewater.
Fair enough. All right. Well, thank you everyone for joining in the discussion today. And like I said, I think we'll aim to have our next effects group meeting in August, just because July is typically a time when people are away, and we also have the teleconference around the same time. So if anyone has papers they want to send, I like the suggestion of thinking about PFAS. Contamination in plastics, especially from the perspective of additives to clothing or to other plastic types. And so look around. If anyone stumbles across a really good paper on that topic, send it our way. So happy almost summer, everyone, and see you later.
Thanks, everyone.
Same to you. Bye.
Bye.